everyone, and welcome to today's webinar, Unlocking Data Insights on Benchling for Novel RNA Therapeutic Delivery Methods. I'm Christy Jewell of Labroots, and I'll be your moderator for today's event. I'd like to introduce you to our speakers for today's webinar. Adrian Simpson, Associate Scientist, HDT Bio, and he is joined by Dr. Vega Shah, Senior Product Marketing Manager, Benchling. For complete biographies on our speakers, please visit the Speaker Bio tab at the left of your screen. Today's presentation is interactive, and we encourage you to participate by submitting as many questions as you want at any time you want during the presentation. To do so, simply type them into the Ask a Question box and click Submit. We'll answer as many of your questions as we have time for. Any questions for Vega will be answered following the presentation, and any questions directed for Adrian, they will be answered via the email address you provided at the time of registration. You may also submit any technical issues here as well if you have any trouble seeing or hearing the presentation. I would also like to bring your attention to the Resources tab at the upper right of your screen. Explore your options in managing RNA data using Benchling here. Vega, Adrian, welcome. You may now begin your presentation.
Thank you for that introduction and for joining us today for this webinar. A quick look at the agenda. We will kick this off today with an introduction to Benchling's R&D cloud. We heard a little bit about it, but I'll get a little deeper into what Benchling offers to research groups. And then I'll pass it on to Adrian to talk about vaccine formulation at HDT Bio. He's going to get into the data architecture used to capture formulation data and some of the challenges faced by HDT Bio on QC and stability. And then finally, he's going to wrap it up by talking about how they manage all of this information on Benchling. We will end this webinar with a Q&A. So our software capabilities at Benchling empower scientists to work mostly in one place for all their research and development needs. R&D teams using Benchling software or applications are able to streamline experimental data capture, do biomolecule design, registration, and in vivo data management all in a single platform. R&D teams also use Benchling for end-to-end tracking of samples so that they can iterate on their next steps without redundancies. We have recently also added workflow management and integration with lab instruments. Benchling is a cloud-native platform, which means it comes with a unified data model, and it has access control and helps research groups meet some regulatory compliance needs by having things like an audit history. We've also observed that in a research lab, scientists tend to use a variety of different platforms that make up an ecosystem. So we have made the platform very extensible. There are open APIs to allow you to connect with other software. There's codeless configuration to customize it to your needs. And Adrian will talk a little bit about this during his presentation about how HDT Bio is using Benchling. And then finally, we have a data warehouse that allows you to capture very large data sets that are common for very high throughput experimentation. Okay, now over to Adrian.
Hi, my name is Adrian Simpson, and I'm an associate scientist on the formulations team here at HDT Bio. Today, I'll be talking to you about how we develop vaccine formulations for the delivery of self-amplifying replicon RNA. First, a little background on our company. HDT Bio was founded in 2018 with the primary directive of developing immune therapeutics for all. Of course, as we know, when 2019 came around, we were immediately faced with the largest pandemic the world has ever seen, which luckily we were well positioned to respond to. We were able to rapidly develop a self-amplifying COVID vaccine, which utilizes a novel delivery mechanism, which I'll elaborate on in the coming slides. To date, our vaccine candidate has undergone multiple clinical trials in several different countries and has an excellent safety profile. Additionally, our company has vaccine projects focused on several other disease targets, as well as cancer therapeutics and immune agonists. Our vaccine candidate, which is known as Lion, consists of an oil and water emulsion that uses a cationic lipid incorporated into an emulsion to bind RNA via charge-charge interactions. The oil core of the particle is stabilized using surfactants and decorated with the head groups of the cationic lipid. This allows for the negatively charged RNA to associate with the emulsion. You can see in the graphic to the right. The emulsion then acts as a delivery vehicle for the emulsion RNA complex as it is delivered to the cell. The RNA is then able to replicate and produce the encoded protein. This complexing method has several distinct advantages as compared to other delivery mechanisms, such as LNPs, specifically because our formulation forms a shelf-stable emulsion without the addition of RNA. The Lion platform is able to be mixed with RNA at bedside, as opposed to an LNP that would need to be mixed with an LNP at manufacture. Bedside mixing is advantageous because the RNA is the least stable component of the vaccine, and therefore Lion can be stockpiled and stored independently of RNA. This eliminates the need for the cold chain during distribution and greatly increases vaccine component shelf life. Due to the formulation of our vaccine, we also see an improved safety profile, especially when compared to other replicating RNA vaccine candidates, which to date have had significant adverse effects in clinical trials. Self-amplifying RNA, for those that are unfamiliar, utilizes viral replication mechanisms, not only to produce the desired protein, but also to create additional copies of the RNA, which has several advantages over the more commonly used mRNA. Unlocking self-amplifying RNA is also advantageous for our platform, as we are able to show dose-sparing effects, since the replicon is able to increase protein expression through replication. To help you better understand the work that I do at HDT, I will walk you through the manufacturing process we use to make our emulsion formulations. The manufacturing process begins by separately weighing out the hydrophobic and hydrophilic components of the formulation. The oil phase generally consists of a liquid oil component, a lipophilic surfactant, any stabilizing agents that we're incorporating, and a cationic lipid. The aqueous phase generally consists of the hydrophilic surfactants and any buffering components. Both the oil and aqueous phases are then heated in a bath sonicator above the melting temperature of any of the solids. Once melted, the oil and aqueous phases are combined and then are high shear mixed to create a crude emulsion. The crude emulsion is then added to our microfluidic device, which utilizes pressures up to 30,000 psi, to push the crude emulsion through the interaction chamber. This causes shearing forces on the larger oil droplets, which reduce in size to form stable nanoemulsions. From there, the formulation may be sterile filtered and is then ready to use to deliver RNA. Our standard emulsion is around 60 nanometers in diameter and is shelf stable at 5C for around 18 months. This process is advantageous because it is incredibly scalable. On our single machine that we have, this microfluidic M110P, we can manufacture between 100 mils at kind of minimum up to around a liter in a single run. There are other instruments that would allow the manufacture of volumes as low to 3 to 5 mils, as well as I've seen hundreds of mils on a GMP scale, all utilizing the same basic principle. One of my principal responsibilities on the formulation team here at HDT Bio is the development of novel formulations for various applications and delivery routes. Since our formulations consist of a number of components, each of whose chemistry may be changed, as well as the ratios between these components being a significant factor in the formulation's physicochemical attributes, as well as its performance in vivo and in vitro, We often find that it's useful to utilize design of experiment principles to help understand the correlation between factors. Briefly, design of experiments is essentially applied statistics that deals with planning, conducting, analyzing, and interpreting controlled tests to evaluate factors that describe the value of parameters or groups of parameters. In so doing, DOE gives us two powerful tools. Firstly, it defines and often reduces the number of samples, in this case formulations, that need to be tested. And it also helps by showing trends between variables, also including so-called higher order relationships that are often difficult to observe and quantify normally, such as logarithmic or polynomial relationships. Here I have an example of experimental outputs that we were able to produce taking a DOE approach. The chart on the right represents a multivariate analysis where the X and Y axes are various inputs and results. These are then plotted to compare trends. In general, we are looking for something that can be fit with a trend line to show that two factors correlate with one another. Or in other words, when one variable changes, the input result or input, the results are positively or negatively affected by that change. So in this example, we see both strong positive and negative correlations. We also see a good number of factors that do not show any correlation. To further explain, each point here represents a different formulation that was made. This was manufactured using the DOE principles to explore the correlation between the factors that we are attempting to test. The results then help us look at how these changes affect the overall performance of the formulation. Statistical analysis of these factors yields correlation results and significance probabilities. See the table to the right. The color of the bar represents a negative or positive correlation, so negative in red and positive in blue. The size of the bar represents the slope of the correlation fit. So you can see something that's steeper is going to have a higher bar, something that's shallower is going to have a lower bar. And obviously anything that is kind of in the middle is probably a trend line that shows no significance, like some of the larger ellipses you see there on the left. Any correlations that are statistically significant are highlighted in green. This automatic processing significantly reduces analysis time and especially allows for deeper interrogation of the data. Of course, to be able to interpret this data in depth, it is also critical that the data is organized and accessible. And this is where I found Benchling to be an excellent tool. So I'd like to go over the data architecture that we put in place to manage our research here at HDT. So when I started at HDT, we were near the beginning of the design of our Benchling implementation. And so I was able, alongside with my coworker Kyle and our Benchling implementation expert, we were able to design our tenant architecture to best capture our data. At this point, I've experienced relatively good data management in my career when we had database tools at one of my previous jobs, as well as much more lackluster data management when we lacked these tools, which made analysis much more difficult. So I was determined to make sure the data architecture was well set up, because as we know, it's very difficult to fix a data set once it's already been utilized as compared to starting off on the right foot. So I thought it was pretty critical for my team to start off at the right point. To that point, I would like to walk you through the design to help you understand how we were able to capture our data and surface it in manageable ways. To start with, we generate a formulation raw material entity. This represents a chemical component of a formulation, so something like a helper lipid, like DSPC, or a surfactant, like tween. The entity itself is relatively simple, but it's the building blocks upon which we build every formulation. From there, we have a parent-child link to the formulation raw material lot. You can think of this as a representation of the physical chemical in the lab, while the formulation raw material is like the conceptual chemical structure. Then a formulation entity would be created. The formulation entity you can think of as the recipe for a formulation. It primarily consists of formulation raw material entities with assigned concentrations, as well as some metadata about the formulation itself. Next, a formulation lot entity is created, which again acts as a digital representation of the physical entity in the lab. It has both a parent-child link to the formulation, as well as it has calculated fields that surface the recorded formulation raw material entities on the formulation page. This allows a user to easily input the formulation raw material lots that are being used in lab to create the formulation itself. Again, the formulation lot also has metadata associated with it, data like manufacturing lot size and manufacture date. From there, a lot may be used to generate formulated complexes by mixing it with an RNA. I've kind of shorthanded the RNA here coming from the side. The RNA entity structure is much the same as the formulation lot structure, so I won't go into detail here. Then both formulation lots and formulated complexes are commonly used to record QC or experimental data against. Using a number of different data types, anywhere from pH and particle size, which are more common for formulation lots, or transfection efficacy in vitro or in vivo, which is more common for formulated complexes. The next large challenge to overcome when we were designing our database architecture was the large breadth of data we aim to encapsulate, as well as the management of tasks and inventories. One of the potentially more unique problems we faced was the generation and management of stability tasks. Our formulations were put on long-term stability programs to help understand the effects of chemical changes on the stability of our formulations. Lots that are put on stability are filed and stored at 5 degrees, 25 degrees, 42 degrees, all Celsius, obviously. Each sample is then held at the assigned temperature until a specified time point is reached. They are then checked for particle size and component concentration by DLS, or dynamic light scattering, for particle size, and HPLC for chemical concentration at the various time points. You can see our general schedule on the right-hand side here: 1 month, 3 month, 6 month, 9 months, 12 months, 18, 24, and 12 months, so on from there on. As I mentioned, our formulations generally are stable for upwards of 18 months, and we like to see them go all the way to failure. So even if they're not in the best shape, we'll keep them on a little bit longer to understand that failure trajectory. Another problem that I assume all labs face is the dispersion of data. Between experiments, varied loss in formulation entities, as well as between projects and grants that we work with, our data is bucketed and distributed in many different ways. This can be especially aggravating when it comes to writing final reports, especially for long projects where the data may be scattered amongst a long series of experiments, and without good tagging and management, it can be a nightmare to extract. Finally, another significant issue we were looking to address, especially with our formulation stability program, was the management of formulation and material inventory, which I'm sure anyone listening in a lab is aware that can be a major issue, especially if not implemented with care. Luckily, using a combination of pre-built benching tools and a few unconventional applications within Insights, and of course, a whole lot of meetings and discussions, my team was able to implement solutions to most of the aforementioned problems. It is not to say that we solved all the lab problems out there, but we were able to implement functional and relatively low effort, efficient workflows for our largest and most common problems in the lab. The most significant issue my team faced was generating tasks for our stability program. The issue we faced is monitoring hundreds of formulations for their stability tasks and notifying the team when they needed to run certain assays. It's a tall order for anything short of a dedicated customized system. What my coworker and I were able to work up is a set of insights blocks, which individually query the formulation lot database for the manufacture dates and surface formulations that fall within specified time windows. So to the end user of the dashboard has a series of blocks which represent stability time points that have been put in place. The user then simply updates to the test date to the current date as a variable at the top of the dashboard. It will then surface all the formulations which have stability tasks within the given period. So if I make a lot today and we come back a month later, they can input the day, whatever it is a month later, and my lot that I made today will show up having a stability time point at the 1 month time point. Then those tasks can easily be added to work lists to import into our stability entry template. We will also surface a field which is part of the formulation lot entity schema, which utilizes a dropdown to select the stability temperature that the original user will first specify, and then it also allows our operator for the stability program to update the temperature list when a given formulation fails at a certain temperature. Another useful feature that's been widely adopted by our lab is zebra printing templates, which utilizes Benchling's built-in inventory system, which is stellar, and I'm sure most of you are familiar with, so I kind of won't harp on about it here. But a stability program, of course, is only as useful as the samples that make it up. So being able to easily find and run these samples for analysis is a critical component of the program being successful. So it was critical for my team to be able to translate the location information in the computer to the real world, which is where the printed templates have been invaluable, as we're able to quickly generate informative labels with minimal effort. Being able to rapidly aggregate data sets is another unsung hero of the database model for my team. Going back to the data architecture I talked about earlier, this is where the parent-child link proves invaluable, as it is often the case that we want to look at the results of all the lots of a formulation at once rather than looking at the individual lots. And it can be painful to aggregate that data if it's not in an easily accessible format. To this point, we were able to use the parent-child link of our formulation dashboard, which aggregates QC data and experimental results recorded against all the lots of a specified formulation. This has been a lifesaver when my boss comes to me last minute asking for a report and when I don't have the data pre-prepared. Similarly, my coworker was able to generate insights dashboards, which can accept a broad list of formulations and allow for rapid data aggregation when required. This utility is difficult to replicate in my experience and has been a valuable tool for modern day data analytics, such as queries generated from the discoveries from a DOE. I recently used this. I was able to down select formulations from a previous DOE to try to input into a new DOE, and I was able to aggregate all that data very rapidly just from the list that I generated. And with that kind of final statement, I've concluded all the planned topics for this webinar, so I hope you found this information useful, and please stick around for questions. Thank you.
Thanks, Adrian, for that great overview of HDT Bio's work. And thank you for the audience for attending this webinar. We will now open it up for questions.
Thank you, Adrian, for that informative and applicable presentation. It is now time for the live Q&A portion of our webinar. And now to our audience, if you have any questions you would like to ask, please do so now. Just type them into the Ask a Question box and click Submit. And we will answer as many of your questions as we have time for. As a reminder, any questions submitted for Adrian, they will be answered via the email address you provided at the time of registration. Okay, so Vega, we have some great questions already coming in. Let's start with this one. What are some specific features or offerings in the Benchling platform that can support RNA research?
Yeah, great question. And I think Adrian covered some of this, but I can expand on that as well. So benchling was always designed to be biologically aware across the platform. What that means is it's specifically designed to take entities like RNA and create information around it. You can design RNA sequences and oligos very efficiently on this platform. You can store RNA as an entity in the registry and make it searchable and traceable. You can visualize sequence features and chemical modifications on RNA alongside experimental data or assay data. When you enter new entities, there's an automatic uniqueness check that allows you to sort of standardize and have correct information going into your registry. You can execute one or more chemical modifications now easily across the entire sequence with the find and replace feature. We also let people create a monomer library to manage modifications if they want. We have an advanced search that lets you find part or the full RNA sequence with or without chemical modifications. This customizable data model that allows you to connect and track lineage of stored entities and samples. And then Adrian talks a bit about the Insight dashboard. That's the part of Benchling that allows you to aggregate all the data. So for example, you could search for one particular RNA sequence and quickly dive deeper into what experiments have been done related to that, or how many samples do you have in the inventory related to that. So there's this ability to share results and data with full experimental context at all times.
Excellent. Thank you, Vega. And will Benchling support circular RNA at some point?
Yeah, good question. So we don't at this time, but this is, we've had this request quite a few times, and also related to cyclic peptides. So this is something the product team is researching now, and we'll probably see more movement on it next year.
Very good. And now this question has come in several times. Does Benchling support working with CROs and CDMOs?
Yeah, great question. So we do have quite a few users that work with either CROs or just external research partners for their therapeutic work. There are three flavors of how you can interact with CROs. The first one is you can onboard a CRO as a user on your Benchling tenant. What this allows for is they can enter any assays or research they've done directly into that tenant, and then scientists at your organization will have direct access to that experiment, but also the experimental context in real time. This is probably the most sort of involved version. The second one is you can collaborate with CROs, where Benchling is used as a drop-off point for data. So the CRO can log in, they can drop in the file containing the data, and then scientists at your organization can just pick it up as a notebook entry. And then the last one, which requires the least amount of sort of implementation, is where the CRO sends you the data via a third-party application or email. You grab it from your email, and then you can save it within an experiment, attach it as a file, or even transport it as a structured table.
Excellent. Now, we've heard artificial intelligence and machine learning all over the news. Is there, well, we're hearing where, there's a connection in with drug discovery. Does Benchling have any AI tools?
Yeah, so this is something we're researching. It's an ongoing thing at Benchling. We're very excited about AI/ML tools being used for drug discovery and even for practical purposes in doing experimental work. We recently launched earlier this year AlphaFold 2, which is DeepMind's open source AI algorithm that can predict 3D protein structures. So this is now available within Benchling's molecular toolkit, and anyone can use it to, have a amino acid query and predict its 3D protein structure. One example how this could be used for RNA research is to predict the structure of RNA binding proteins. This is only one example. We're currently researching many other options. So as Benchling as a platform develops, we'll definitely see more AI and ML tools coming out.
Excellent. Thank you, Vega. And it looks like that's all the time we have for today for our questions. Do you have any final comments for our audience?
No, thank you for being here and really excited to see these great questions.
Very good. Thank you. And to our audience, if you do have any additional questions or would like any more information about Benchling, please type them into the Ask a Question box, and you can do that during the on-demand period as well. So I do want to thank again Adrian Simpson of HDT Bio for his time and valuable insight today, and to Dr. Vega Shah for this informative Q&A session. We'd also like to thank our sponsor, Benchling, and Labroots for helping to bring this educational webcast to you. Now, I do want to thank the audience for joining us today and for their interesting questions. Again, as a reminder, any questions for Adrian that were submitted and those that are submitted during the on-demand period, they will be addressed by our speakers via the contact information you provided at the time of registration. Today's webcast can be viewed on demand, and Labroots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, everyone, thanks for joining us and have a great day.
Thank you. Bye.